Hello everybody, now we're going to move on to insulin signaling at the muscle. So now that we've learned how insulin is secreted from the beta cell, now we're going to learn about what that insulin does when it reaches its target tissue. And the first tissue we're going to talk about is skeletal muscle, because it's a large tissue that takes up a lot of glucose for energy needs, and it's important to understand how this process works. We're first going to start with a video that will give us a good overview, and then we'll talk about the pathway. All right, so this is a video that's going to give us an overview of the insulin receptor and the insulin signaling cascade, and then again we'll go over it in just a moment.
Hi, this is Mark from the MSOE Center for Biomolecular Modeling. The following animation will show the molecular mechanism whereby insulin triggers the uptake of glucose from your bloodstream. Soon after a meal, the digestive system begins to break down the food you have eaten. The carbohydrates in this food are broken down into a simple sugar called glucose. As this glucose is absorbed from your digestive system into your bloodstream, your blood sugar levels increase. Your circulatory system then carries the glucose to muscle cells throughout your body, where it is used to generate energy. And here's the important part. In order to get that glucose that's now in your bloodstream into your muscle cells, you need to have insulin present to trigger that uptake event. Insulin is a small protein hormone produced by your pancreas. As the concentration of glucose in your bloodstream rises, your pancreas senses this increase and is stimulated to release insulin into the bloodstream. The newly released insulin plays a key role in regulating the concentration of glucose in your blood, a process known as glucose homeostasis. The insulin that is now released into your bloodstream binds to the extracellular domain of receptor proteins found on the surface of liver, muscle, and fat cells. This binding triggers the autophosphorylation of the intracellular domains, which in turn phosphorylate a specific substrate signaling protein. This protein then phosphorylates other downhill signaling proteins, leading to an amplification of the signal at each step. This overall signaling process is known as a signal transduction cascade. One important consequence of this signal cascade is the movement of glucose transport proteins, called Gluts, towards the cell surface. As these storage vesicles fuse with the cell membrane, the number of Gluts present on the surface of the cells increase, allowing the glucose to enter the cell. As a result, the glucose concentration in the bloodstream decreases. The glucose is now inside the cell, where it can be metabolized to generate the energy in the form of ATP that is needed in all of your cells. And that is how insulin helps the sugar you eat turn into the muscle that you use.
All right, so that's a video of an overview of the insulin signaling cascade. And so let's take a moment and let's talk about it. All right, so this is the insulin receptor that is important, that gets this all started with the insulin signaling cascade. And it's important to understand that it has two subunits. It has an alpha subunit that's in the blood, and here's the cell membrane, and then it has a beta subunit that's in the tissue itself. So you have an alpha subunit and then a beta subunit. And again, here is the alpha subunit, and here is the beta subunit, and here is the plasma membrane. And how this works is that insulin is going to bind to the alpha subunit, and then that is going to cause an autophosphorylation of the beta subunit. And autophosphorylation simply means that a phosphate is added to something. In this case, a phosphate is added to the tyrosine. And tyrosine, if you remember back to your chemistry days, is abbreviated TYR. So you have a phosphorylation, and the phosphate is coming from ATP that's in the tissue itself. So essentially ATP will come in, add a phosphate to tyrosine on the beta subunit of the insulin receptor, and that's going to then continue the insulin signaling cascade. So here we have the insulin effects on glucose transport and skeletal muscle. So to orient you, this right here is the blood. Here's the cell membrane of the skeletal muscle, and then inside here is the skeletal muscle. And what we have here is the glucose molecules that are circulating in the blood, and herein is the insulin receptor. So let's talk about how this insulin signaling cascade works in order to get glucose into the cell. So the first step is you have insulin from the pancreas is released in response to an elevation of blood sugar, blood glucose. That insulin will travel out of the pancreas to the skeletal muscle, and the insulin will attach to the alpha subunit of the insulin receptor. And then this will cause a series of phosphorylations or autophosphorylation, which is the same thing for our purposes. This is going to cause a autophosphorylation of tyrosine. And notice that the Y here represents tyrosine. That's just an abbreviation in chemistry for tyrosine. This is going to cause a tyrosine autophosphorylation, which just basically means, again, the phosphate coming from ATP attaches to the tyrosine that's found on the beta subunit. Well, this is going to cause a series of autophosphorylations, the cascade. The first is going to be an IRS 1 and 2. That's not IRS 1/2, but IRS 1 and 2. So the insulin receptor substrate 1 and substrate 2. And that's going to add a phosphate to the tyrosine that's found on the IRS 1 and 2. Well, this is going to then cause a tyrosine phosphorylation of PI3 kinase. You can see the word here. It's a very long word, but it's abbreviated PI3 kinase. Then this is going to cause serine phosphorylation of AKT, so serine abbreviated with the S, and that just simply means that the phosphate is going to attach to the serine amino acid on AKT. And then through a series of events that we won't talk about, but it's going to then activate these GLUT4 transporters that are going to remain in the center of the skeletal muscle, waiting for a signal to cause them to move to the cell membrane. So they remain here until they finally receive a signal to move to the cell membrane. Once they receive that signal, the GLUT4s in skeletal muscle will move and make their way to the cell membrane and embed into this cell membrane. And then, acting like a channel, the glucose will make its way through the GLUT4. So once it goes through the glucose goes through the GLUT4, it's going to come into the cell and they either undergo glycolysis, which is the breakdown of glucose to form ATP, or glycogenesis, which is simply making glucose into glycogen to be used for later energy needs. So these are the steps at which glucose enters the skeletal muscle. Notice how insulin never entered the skeletal muscle. It only remains in the blood attached to the alpha subunit of the insulin receptor. But then there's a series of autophosphorylations that then carries it through to the GLUT4, and then finally the GLUT4 will make its way to the cell membrane, allowing glucose to move through the GLUT4 and into the cell. By doing so, glucose enters the cell, leaves this blood, and therefore your glucose levels will drop. So take a moment here and review this. We're going to talk about it in just a video in just a moment, but make sure that you have a good understanding. We'll come back to this in just a moment. Here are the steps that we just talked about in this pathway. You can find these in your notes as well. And here is a video we're going to watch now that kind of gives another feature or another view of the insulin signaling cascade. All right, so this video is going to give another example of the insulin signaling pathway. So pay close attention to it, review it several times to make sure you have a good understanding of it.
A biological individual consists of multiple organs with specialized functions. For the organism to function properly in its environment, these organs must communicate. This communication often involves a signal sent from one location to another that instructs the second organ about the status of some cellular feature. Glucose is a good example. Glucose is a critical product of digestion. It is an essential energy source for cellular metabolism. This energy is produced when glucose is used as a substrate for glycolysis and then the Krebs or citric acid cycle. Following the digestion of food, higher levels of glucose circulate through the bloodstream, where it enters different cell types. In muscle cells, glucose is readily used to produce energy and is also stored as glycogen, a secondary short-term energy source. In fat cells, glucose is used for triglyceride production and acts as an important energy reserve molecule. Here, we will illustrate the signaling pathway that occurs when glucose is at high levels. This pathway involves multiple proteins and signaling events. This is termed cytoplasmic signaling.
So here, just to give you an idea that this is the insulin receptor. Here's the alpha subunit, here's the beta subunit, and here that you notice here actually is the ATP with the 1, 2, 3 triphosphates. And there you're going to see that again the ATP is coming in and adding its phosphate, and you'll notice that that sends a signal to one protein down to the next protein, et cetera, et cetera.
Different types of cells perform similar signaling steps in response to changes in their environment. In the protein recycling animation, we see a group of storage vesicles enriched with GLUT4 proteins continuously recycling from the cell membrane to an inactive location in the cytosol. GLUT4 is a protein that facilitates the movement of glucose into the cell. When high levels of glucose are detected by beta cells in the pancreas, insulin is released by the cells. The insulin circulates through the bloodstream until it binds to an insulin receptor embedded in the cell membrane of a muscle, fat, or brain cell. Once the insulin binds to the receptor, phosphate groups are added to the intracellular domain of the receptor. Since the receptor itself adds the phosphate groups, the process is called autophosphorylation. This phosphorylation event sets off a cascade of molecular events. The activated receptor protein then adds a phosphate group to another closely associated protein. This effectively passes the signal from the receptor to the next step in the signal pathway. Proteins that add phosphate groups to another protein are called kinases. Kinases are often components of signal pathways, and phosphorylation is an important component in the transmission of a signal from one compartment to another. In this system, the signal corresponds to the level of blood glucose and is transmitted from outside to inside the cell. Next, we see a large pool of molecules that are embedded in the membrane, also being phosphorylated. Other proteins are then in turn phosphorylated, further transmitting the first extracellular signal that was originally sent from outside the cell membrane. So how does this affect the uptake of glucose? As we mentioned before, GLUT4 is a glucose transporter, and GLUT4 storage vesicles are held in a recycling state near the cell membrane. The vesicles are held mostly in this region because the Rab proteins that interact with the motor proteins necessary to move the vesicles to the membrane are in an inactive state. The final step in this signal pathway involves the phosphorylation of a protein that prevents the Rab proteins from interacting with the vesicles. When the Rab proteins are no longer inhibited, the storage vesicles can freely merge with the membrane. Once the vesicles have merged, many GLUT4 proteins are embedded in the membrane, and large quantities of glucose can move into the cell. It is the signaling pathway that ensures only the correct molecules will be allowed to enter the target cell.
All right, so this here is, you see those, the GLUT4s, they're embedded in the cell membrane, and you see the glucose that now is coming through into the cell itself. So this hopefully was a good video that gives you an overview. You don't need to understand the RAB proteins that they were talking about towards the end. Just focus on the steps that we talked about in the pathway. So again, make sure that you have a good understanding of the pathway. You can do practice on Blackboard and also compare it to give you a good idea and understand if you have a good understanding of it. Also, you can write the steps in your own words and then compare them to what we have here. Now you notice that as we move forward that in here they talked about GLUT4 transporters that are found in the skeletal muscle. It's important to know that we have a number of GLUT transporters throughout our body. And before we get to that, insulin is one that actually activates these GLUT transporters. And again, as we mentioned in previous slides and videos, insulin is like a key that's going to turn on the glucose transport ignition. So in our case, and what we just saw, it basically activates the GLUT4 transporter. When the key or insulin activates the ignition, the insulin receptor, the engines or glucose transporters that do the work of hauling glucose out of the bloodstream and into the cells are activated. So the idea is that insulin basically is going to activate these GLUT4 transporters to come to the cell membrane to allow glucose to get into the tissue. So this helps get the glucose into the cell in order to get feed the tissue with energy, but also reduces the blood sugar. Well, it's important to know that we have different types of GLUT transporters based on the tissue of the body. We've talked about the GLUT4 transporters in the muscle. They're also located in the fat cells. So what we have here is we have the transporter, GLUTs 1 through 5. You have the major site of expression, which basically means the tissue that you'll find these particular GLUT transporters. And then finally attracted to glucose. What this basically means is what level of glucose does there need to be in the body for these GLUT transporters to be activated and turned on? Well, you'll notice that some are going to be activated, such as GLUT1 that are in the brain, are going to be activated even at low glucose levels. Contrast that with, let's say, the GLUT2 located in the liver and some other cells. They're only going to be activated when glucose levels are high. We have GLUT4, which we talked about, in the muscle when it's going to be activated when glucose levels are moderate. The reason for this different tiering level, the different levels of attractiveness of these GLUT1s based on the glucose levels, is it ensures that tissues that need the glucose the most will get it first, meaning that GLUT1 is going to be activated even at very low glucose levels, blood sugar levels, to ensure that the brain and its nervous system is receiving glucose so it can function properly. Without our nervous system working properly, nothing in our body will work. So it ensures that even at low glucose levels, that the brain and the nervous system is going to receive glucose. Contrast that where at only high levels will the liver, and at moderate levels will the skeletal muscle be receiving glucose. Well, this tiering system ensures that only when glucose levels are high or moderate will these tissues get it, but even at low levels, it ensures that the brain through GLUT1 is going to be receiving glucose to ensure that our body can function. So you have this tiering system based on the glucose levels. And this ensures that the tissues that need it most, even at lower glucose levels, will receive it. And then as glucose levels rise, other tissues will start to receive the glucose as well. So there's a couple that you should be familiar with. In particular, GLUT2 has a low affinity for glucose. In other words, it's the major transporter in the liver and the pancreatic beta cells. And again, this has a low affinity of glucose, meaning that only at high blood sugar levels will GLUT2 be activated. And this reason for this ensures that insulin is secreted only when blood glucose levels are high. It also prevents hepatic uptake of glucose when levels are low, such as during fasting periods, basal periods. So as we just talked about, GLUT2 is only going to be activated when glucose levels are high. Otherwise, the other tissues are going to be taking in the glucose. So for example, GLUT4 is found in two major targets, the muscle and the adipose tissue. And again, these are only going to be activated when blood sugars are about moderate levels. So generally speaking, after a meal. But the idea with this tiering system is it ensures that the brain receives glucose first, followed by the adipose tissue, skeletal muscle, and then finally, when only levels are very, very high, will the liver and the pancreas, etc., receive glucose. So we have this, again, this tiering system that protects our body and ensures that tissues that need the glucose most, even at low levels, will get it first, followed by these other tissues as your blood glucose levels rise. So what is the role of insulin then? We've talked a little bit about how the role of insulin with the pathway of the skeletal muscle, but let's talk a little bit more in detail because this is going to be very, very important as we especially go into lesson number 5. But the key thing to understand here is that the major function of insulin is to promote storage of ingested nutrients. So think of it as a storage of nutrients. At the same time, the second important aspect of insulin is to prevent breakdown of nutrients. So there's two things to understand with insulin. It increases storage of nutrients and it prevents the breakdown of nutrients. So for example, in the liver, if you see the bold are the ones that you really need to know. The liver it promotes glucose storage as glycogen. So again, insulin causes glucose to be taken up in the liver. In other words, it's causing the glucose to be stored as glycogen in the liver. At the same time, it inhibits glycogen breakdown. So again, the two things that insulin does, increases storage of nutrients and prevents the breakdown of nutrients. Now we're talking about specific nutrients, glucose, and that's going to be found in skeletal muscle and liver. And then we're also going to talk about triglycerides and adipose tissue, which we'll get to in just a moment. But just know those two things, that insulin promotes glucose storage or triglyceride storage, and it prevents the breakdown of these nutrients. Okay, so in the liver, insulin promotes glucose storage, and it gets stored. Glucose gets stored as glycogen. At the same time, it prevents glycogen breakdown. In the skeletal muscle, it increases glucose transport, in other words, getting glucose into the cell, and it increases glycogen synthesis. So skeletal muscle, basically again, it increases muscle or glucose storage into the skeletal muscle. In adipose tissue increases triglyceride storage and also promotes uptake of fatty acids. So this is important. So adipose tissue, insulin is going to increase triglyceride storage, and it's going to promote uptake of fatty acids. And it also is going to prevent the breakdown of triglycerides. So again, insulin increases storage of nutrients and prevents the breakdown of nutrients. So when we're talking about adipose tissue, it's going to increase triglycerides and fatty acid storage, and it prevents the breakdown of triglycerides and fatty acids. So again, those are the important things to know with insulin. Increases the storage of nutrients and prevents the breakdown of nutrients. So these are the important metabolic effects of insulin that you should be aware of. Here we have, again, just to illustrate this, about the insulin's effect. When you have insulin released by the pancreas, again, it's going to prevent the glucose or liver from secreting glucose into blood. That's what this little line means, prevents glucose from being released by the liver into the blood. This also causes glucose to be taken up by the skeletal muscle, and also it causes glucose and fatty acids to be taken up into the adipose tissue. So The idea remember with insulin is that it causes an increased storage of nutrients, glucose and triglycerides, but prevents the breakdown of these as well. So what happens in a type 2 diabetic? This is going to be very important. This sets us up for lesson 5. So make sure we're about to talk about something that's very, very important. Here, when you have insulin resistance and type 2 diabetes, it's important to understand what happens. Well, in the liver, glucose will not be taken up, and glycogen will be broken down and secreted into the blood. And as a result, when glucose isn't taken up and glycogen is broken down by the liver and secreted in the blood, it results in an increase in blood sugar. So this is what happens when the liver is no longer recognizing insulin. The insulin thinks, or the liver thinks, hey, I don't recognize insulin. Insulin's not around, so therefore blood glucose must be low, and therefore I'm going to go ahead and I'm going to break down the glucose or my glycogen secreted into the blood. At the same time, I'm not going to take up that glucose because I don't want to take glucose out of the blood. However, blood sugar is already high, but the liver doesn't know that because it's insulin resistant. So this is one of the major hallmarks of type 2 diabetes, when you have a result in increased blood sugar. Also in skeletal muscle, glucose will not be taken up because the skeletal muscle is now insulin resistant and no longer recognizes insulin anymore, and therefore the skeletal muscle is like, wait, there's no insulin around, therefore I will no longer take up glucose. So it will not go through that pathway that we just talked about. At the same time, you have adipose tissue. Well, adipose tissue, the role of insulin, as we just talked about, is to take up triglycerides and fatty acids. However, when adipose tissue is insulin resistant, it no longer takes up those triglycerides and fatty acids. They will not be taken up, and therefore they'll remain in the blood. At the same time, because insulin is supposed to suppress the breakdown of triglycerides and fatty acids, and because the adipose tissue is now insulin resistant, triglycerides and fatty acids will now be broken down and secreted into the blood because insulin is no longer preventing the adipose tissue from breaking down triglycerides and fatty acids. So in other words, the adipose tissue is insulin resistant, and therefore it no longer takes up triglycerides and fatty acids. And at the same time, because it's insulin resistant, it continues to break down the triglycerides and the fatty acids and secrete them into the blood. The result is you have an increase in triglycerides and fatty acids in the blood. And that is going to be a hallmark of a lot of the comorbidities and the problems that are going to be associated with insulin resistance and type 2 diabetes that we're going to get to in lesson number 5. So make sure that you review this part of this lesson, making sure that you understand the metabolic effects of insulin, what it should do in a healthy individual, and then understand what is happening in someone who is a type 2 diabetic or insulin resistant, because this is a hallmark of insulin resistance, and we're going to talk a lot more about this in lesson number 5. All right, so this concludes this part of lesson number 4. Make sure you understand the insulin signaling cascade of the skeletal muscle, the GLUT proteins, and what is the role of insulin in the liver, skeletal muscle, adipose tissue.